I'd like to welcome everybody to today's eye health seminar series. Today's speaker is Dr. Krista McCoy, who's pretty close by. She's up at Harbor Branch. Before she gets started, I'd like to give a little bit of background. So Krista received a Bachelor of Science degree from Old Dominion University, Master of Science from James Madison University, and her Ph.D. from University of Florida. She has broad training in ecological development, toxicology, and behavior. As a postdoc scholar, she was awarded an NRS fellowship from NIH. And then in 2013, she joined the faculty at East Carolina University. She had an active NIH-funded research program there, mentored graduate and undergraduate students in research. She was tenured and was a founding member and president of the Eastern North Carolina chapter of Graduate Women in Science. This summer, Dr. McCoy joined the faculty at Harbor Branch Oceanographic Institute as an associate research professor. And she's currently funded through the HBOI Foundation and is also an active and funded member of the Florida Center for Coastal and Human Health. Her current research focuses on understanding how we prevent common disorders induced by exposure to environmental pollutants. So with that, Krista, it's all yours.
Can you see my slides okay, Greg?
Yes, I can see your PowerPoint.
Okay. So thank you for coming today and seeing my talk. I want to take you on a little journey of the past 20 years that has brought me really to this idea that we need to change our perspective on the way that we study environmental contaminants and pollutants. So there are thousands and thousands of synthetic chemicals in use currently, and only about 2% have been evaluated for human health effects. And just over about half of those that have been evaluated show the ability to alter the endocrine system. And these chemicals are called endocrine disrupting chemicals, and they're pervasive. They're literally all over. They're in our water, they're in the air. We intentionally spray these chemicals on our food. We wrap our children in blankets that are embedded with endocrine disrupting chemical flame retardants. We give our babies plastics to chew on that have estrogenic chemicals in them that leach out. We use so many plastics that they are literally creating islands in the ocean of plastics. And not surprisingly, given all of that, we have these chemicals in our bodies. And so most of us have different types of endocrine disrupting chemicals circulating in our systems all the time. And this is just a graph that shows different flame retardant chemicals. And the white is the portion of the population that is not exposed or is exposed to levels that are non-detectable. Many pesticides are known to disrupt the endocrine system. And so I became interested in these years ago because the route of exposure seemed pretty direct to me. In addition, research was coming out showing that some of the pesticides used around in South Florida altered reproductive development of amphibians. Oops, sorry. And so my dissertation work was conducted in South Florida, and it aimed to understand how agricultural land use and the pesticides that they use there affected reproductive systems of wildlife. And I went to 5 different study sites over a few years and collected animals. And the animals that I focused on are the giant toads. So some people know them by the name cane toads or bufo toads. And so for studying the endocrine system, these toads are actually really ideal for lots of reasons. But one of the main reasons that I focused on them is because their sex hormones cause differences in their appearance. And so this, I just want to put my pointer on, is a female. And so you can see that she has mottled coloration on her back. And this is a male. He's not mottled. And so they have visual tags that can be used to assess hormone disruption. You can just look at them and see if that their sex hormones are functioning or not. So at the agricultural sites, the males and females don't quite look like this. They look like this. So male toads at agricultural sites look like females. And when we dissect these animals and look at their reproductive organs, the females have ovaries that are that can be yolking up eggs, as this one depicts. The yolk in this species is black. And males normally have testes, and that's this conical shaped organ here. This is the kidney, just for reference. The kidney is here for the female. Animals at the agricultural sites, in many cases, were intersex. So they had both ovaries and testes in the same organism. And this is not normal for this species or for amphibians in general. Some fish are sequential hermaphrodites or simultaneous hermaphrodites, but that is not true for amphibians. And so when I compared my different sites based on how much agricultural land use was in the region where the frogs lived, I found that the individuals that were intersex with both testes and ovaries increased with increasing agriculture. And the severity of gonadal abnormalities, as well as the total number of abnormalities, increased with increasing agriculture as well. And when I compared the most agricultural sites to the least agricultural sites, males at the most agricultural sites had decreased testosterone and sperm production. So this brought me to this question of, well, what's happening to the people? And this brought me basically to the end of my dissertation work, and I needed to move on and begin to study things that would help me answer questions that I could that I was interested in, given what I found with the frogs. And so one way that I could have gone with my research is to study the behavior of the toads. So I knew that they had abnormalities in their reproduction or in their reproductive system, but how did their reproductive behavior change? Or how did their reproductive success change? And so those are questions that are interesting, and I would love to study that, and probably will now that I'm here in Florida and can do it easily. But what I decided to do was study the implications of this work on human health. And so we know that endocrine disrupting chemicals correlate with incidence of birth defects in humans. One birth defect that a lot of people don't know about, but is actually extremely common, is a birth defect of the penis called hypospadias. And hypospadias occurs when the urethra or opening does not occur at the distal tip of the penis, but rather somewhere along the shaft or even in the perineum between the penis and the anus. Hypospadias has increased 500% in the last 50 years and affects almost 1% of boys. In some states, it is 1%, but on average, it's about 0.8. And although there are genetic abnormalities that lead to hypospadias, many cases remain undescribed by genetic mutations and are associated with embryonic exposure to pollutants. So I began to study how endocrine disrupting chemicals affect genitalia development, using the mouse as a model for human health. And we've been relatively productive and done some interesting things, and I'll talk about these different papers in a bit more detail in a little while. But what I realized is that knowing the mechanisms through which chemicals induce harm doesn't stop that harm. When I was being trained, I was trained that in order to really do good science and really understand a chemical, we needed to understand its mechanism of toxicity. You couldn't just expose an animal and look at the effect. Oh, look, this chemical causes this effect. You had to try to understand what the mechanism of that causing that effect was. But what I came to realize is that knowing those mechanisms doesn't really help us stop the problem. And so I realized that we needed to change the question. And so rather than asking what mechanisms induce toxicity, I decided that we really needed to start asking what mechanisms induce resilience to toxicity. And in this way, we can develop therapies or supplements that might protect us. So one way to think about this is to think about a dose response curve. And so we use these in toxicology all the time. We run these experiments with a increasing dose of a chemical we're interested in, and we measure the response to those different doses in something that we're interested in, some measure. And what we usually do is focus on the mean. And many times we consider the variation around that mean to be a nuisance. Either it's a mistake, experimental bias, something that's not interesting scientifically. But I started to think about these individuals. Why are they resilient? And is it because of an increase in antioxidants or detoxifying enzymes? And if that's the case, if these individuals have some additional protective enzymes, can we increase those enzymes to increase resilience of the population? And in doing so, decrease that dose response, decrease the potency of the chemical that the animals are exposed to. So this brought me to this idea of studying the mechanisms of resilience. And so the idea is, in the context of birth defects, can we develop a prenatal supplement that will reduce the effects of toxicants on the fetus and protect it from developing these endocrine disrupting chemical induced abnormalities? So we needed a model system, and we developed that in a few different ways. So first, we determined how to reliably induce hypospadias, the birth defect that we were focused on, 100% of the time. And we can do that with a chemical called vinclozolin. It's a fungicide that they spray on coffee and grapes and kiwi and a variety of different crops, not so much in the US, but in other countries. We can induce hypospadias 100% of the time with vinclozolin. And that's important if we're trying to develop a model where we can ask, does this supplement reduce the incidence of hypospadias? If we know for a fact that our exposure regimen would cause the hypospadias, then we can actually ask, are we reducing it? We also needed to be able to quantify the severity of hypospadias in a standardized way. So we developed a standardized scoring system we call MOUSE. It's Mouse Objective Urethral Scoring Evaluation. And so we can determine whether the animal is normal or has different severity scores. And then finally, after doing a lot of dose response curves using a variety of different supplements, we found a supplement that protects against vinclozolin, even at these very high concentrations that induce 100% hypospadias. And so what I'm showing you here is anogenital distance. This is a measure of masculinization, and it's used in mice and in humans. And it is indicative of masculinization that occurred during development. And here, with no sulforaphane, but a lot of vinclozolin, anogenital distance is reduced. And as we increase sulforaphane, we see increased anogenital distance up to about 60 milligrams per kilogram. We can talk about these doses here, but this data, this hump-shaped curve is not surprising, especially for chemicals like sulforaphane. And so we decided to focus on the rest of our work on this 45 milligrams per kilogram dose of sulforaphane. So that's what we use throughout the rest of the work. So we wanted to ask, will sulforaphane reduce the toxic effects of vinclozolin? And we found that it does. So we did a vinclozolin dose response experiment with and without sulforaphane. Without sulforaphane, we get the typical dose response curve that we had validated before. And when we add sulforaphane at 45 mg per kg, we can see that the slope of that line is lower, and the dams no longer have 100% males, 100% of their male babies don't have hypospadias in all cases. So for those individuals that have hypospadias, their severity is also significantly reduced. So that was great. And I think we can reflect on what I just said for a minute. We have a chemical, an environmental contaminant, that we can give to moms, pregnant mice, and cause 100% of their babies to have a birth defect. That's common in humans. And when we supplement with sulforaphane, we can significantly reduce the incidence and severity of hypospadias in those animals receiving those very high doses. But what are the mechanisms that are driving this? So what does sulforaphane do? Well, it activates a transcription factor called NRF2. So when there is no sulforaphane in the system, NRF2 is usually bound to keep 1 and call 3, and it is marked for degradation. And it does not cross into the nucleus. When sulforaphane is provided, it releases NRF2 from this complex and allows it to cross into the nucleus, where it binds to the antioxidant response element and induces hundreds of genes to be expressed. And these are typically antioxidant and detoxification genes. And so sulforaphane, through this mechanism of NRF2, increases proteins that are associated with antioxidant defense and detoxification. So we wanted to know if NRF2 was required for the sulforaphane-induced rescue that we saw. And so we conducted an NRF2 knockout experiment where we exposed heterozygous males, or we mated heterozygote males with heterozygote females. And that way the females were impregnated and carried on average both wild type and knockout babies. So this did 2 things. First of all, it allowed us to compare these genotypes without changing mom's genotype. And it also allowed us to compare, in some cases, brothers that were that were held within the same mom. And so we did this cross of these heterozygote animals, and we genotyped and compared the wild type and knockout individuals after they were exposed to 4 different treatments. And so pregnant dams were exposed to a corn oil control, sulforaphane alone, vinclozolin, and vinclozolin plus sulforaphane. And we found that the sulforaphane and control animals had very low levels of hypospadias severity. Vinclozolin had high severity, which is what we knew would happen. But what was really astounding is that only the individuals with NRF2 responded to the addition of sulforaphane. And so our rescue requires NRF2. So sulforaphane seems to be working through NRF2. And there are multiple organs that could be driving these effects, these protective effects. And so one organ that seems pretty appropriate to think about is mom's liver. So we give mom the chemical and we give her the sulforaphane, and then she has a liver that could upregulate detoxification and protect the babies. And so we have found through proteomics that the dam liver proteome is less variable and more similar to control animals in this sulforaphane plus vinclozolin treatment relative to the vinclozolin corn oil contrast. So that is interesting, but probably not inducing the effects that we find, because the effects that we find with sulforaphane is different in different genotypes of babies. And so moms that had both knockout and wild type individuals would have protected both of those individuals if she was driving the protective effects. But since we see that brothers that are different genotypes have different effects, then we know that this protective effect must be happening in the fetus. And so we began to study the placenta and the liver. So today, I'm just going to focus on the placenta. So we have looked at the transcriptomics and proteomics and the placenta and liver. And today I'll focus on the placentas from the corn oil, sulforaphane, vinclozolin, and vinclozolin sulforaphane treatments. And the data that I'm going to show you is just from the wild type individuals. So we have transcriptomics data, and we can compare each of our treatments back to the corn oil. And I'm going to focus just on these contrasts today. So the first thing I want you to focus on is the vinclozolin relative to corn oil contrast. So these genes show the effects of vinclozolin exposure. So there was 1,002 genes that were affected by vinclozolin exposure. And if we focus on this red circle, these are the effects of sulforaphane plus vinclozolin. And one thing to notice is that the total number of genes is 694. And that's actually a reduction of 30%. So 30% fewer total number, total genes were affected when we gave sulforaphane with the vinclozolin. These genes in the middle, 194, are really interesting. So of the 1,002 genes affected by vinclozolin, only these 194 remain different from the corn oil when we supplement with sulforaphane. So that is an 80% reduction in the effects of vinclozolin. So the expression of these 808 genes were corrected by sulforaphane addition. So they become resilient to vinclozolin when we add sulforaphane. So they're not different from the corn oil in that when we add sulforaphane. So we have two groups of genes that are affected by vinclozolin in this data set that I'm focused on. One group are corrected by sulforaphane, brought back to corn oil levels, and there's another group that are not corrected by sulforaphane. And so I wanted to know if the genes that were most strongly associated with vinclozolin exposure, if those genes were corrected by sulforaphane. And so we might think about these as biomarkers of vinclozolin exposure. And we found that there were 19 genes that were strongly associated with exposure to vinclozolin. And what I mean by that is that they were significantly different from corn oil after correcting for multiple comparisons, which basically means that their variation was low. And so they were consistently affected by vinclozolin. And these genes had a lot of different functions, from immune response, cell growth and cell death, transcription and translation, and cell signaling. And so what I wanted to know is, are these biomarkers of vinclozolin corrected by sulforaphane addition? And we found that in every case, these genes were no longer different when we exposed animals to both vinclozolin and sulforaphane. And so that's interesting. And so that is a one subset of these genes that become resilient to vinclozolin when we add sulforaphane. But how do they become resilient? What's causing them to be able to be resilient? And I believe that these 500 genes here are important in that context. So I think that these are the candidate mechanisms of resilience. These genes are the genes that have novel expression patterns. They were not different from corn oil and vinclozolin only, but they become different when we add sulforaphane. And so these are the genes that we're really starting to try to figure out what's going on with now. What we know from preliminary work is that they're associated with oxidative phosphorylation, cell signaling, development, fetal development, and immune response. And that's one thing that's come up again and again with this work, is that sulforaphane is influencing the immune system, which is really interesting. So sort of immunotoxicology endpoints are affected. So just to summarize what I've told you so far, is that sulforaphane decreases the potency of vinclozolin, this fungicide that we use as a model chemical to induce hypospadias in the mouse. That rescue effect was dependent on NRF2. And the placenta is broadly affected. Sulforaphane drastically reduces vinclozolin-induced gene expression within the placenta. It completely normalized biomarkers of vinclozolin exposure, and the mechanisms of resilience are diverse, more diverse than I thought they would be. And they include oxidative phosphorylation, which maybe isn't so surprising, but also immune responses. So now, how am I using this novel perspective to study mechanisms of resilience here at Harbor Branch? The work that I just presented was really done at East Carolina University prior to moving here. We moved here this past summer. And so I wanted to talk to you a bit about what we're working on at Harbor Branch. So some of the questions that I'm focused on is whether sulforaphane is protective in humans, is it safe, and is it generally protective? So will it work to protect us from multiple kinds of chemicals? And so we need a new model to test if sulforaphane is generally protective. And we focused on toxicant-induced autism spectrum disorder. And this is work that originated through collaboration with a lady at East Carolina University. So autism spectrum disorders are characterized by altered development of neural circuits that manifest early with deficits in social behavior and communication, and increases in repetitive behavior and restricted interests, different interests. Some ASDs we know are associated with exposure to environmental contaminants or pharmaceuticals. We know a lot about autism, both from studying humans, but also from studying rodent models. And so we know that autism presents early. It's a neurodevelopmental disorder. And recent data from the CDC suggests that it's as common as 1 in 54 children in the US. It occurs more often in boys than in girls. In fact, 1 in 34 boys are identified with autism, and only 101 in 144 girls are identified with autism. A lot of people are studying why boys are more affected. But I'm really interested in trying to understand the mechanisms that protect the girls. Another thing that's really interesting that we know is that autism patients do very well with early intervention. And so if we can intervene very early, then they have fewer symptoms and fewer problems. And actually, there are a couple of clinical trials that have shown that Avmacol, which is a sulforaphane supplement, reduces symptoms in autism patients. And so that's really great. But what we really would like to know is, can sulforaphane protect against development of toxicant-induced autism? Can we stop the autism from developing? And so we know that prenatal exposure to the anticonvulsant pharmaceutical valproic acid increases the risk of developing an ASD in humans and leads to autism-like behavior in mice and rats. And this is a well-studied model in rodents to induce autism, so that we can study how it's being caused. And so we leveraged this detailed research that has been done on VPA to use it as a model to study whether or not we could protect the brain from VPA-induced problems with sulforaphane. And so I think, have just gotten funding for an NIH R21. The money is not in the bank yet, so I'm trying not to count my chickens before they hatch, but the program officer said they were in the final stages of funding the project. Don't know what that means. So Karen is going to work with human cortical spheroids to study neural circuit development. And I am going to use the mouse to try to understand behavioral consequences of exposure. And what we're trying to do is identify conserved sulforaphane-induced genetic and cellular mechanisms that protect the brain from valproic acid, and also prevent valproic acid-induced behavioral deficits. So the human cortical spheroids and mice will be exposed to valproic acid, sulforaphane, their combination or control. And Karen is going to use CRISPR to knock out NRF2 in some of her cortical brain spheroids, and then in others, she'll leave them intact. And she's going to test the hypothesis that sulforaphane will prevent VPA-induced neural circuit disruption in these many brains through an NRF2-mediated reduction in oxidative stress. My work is as similar as it could possibly be. We are going to expose mice during a very specific time that is defined by previous studies that use valproic acid to induce autism-like behavior in the mouse. Prior to VPA exposure, we're going to start feeding the animals sulforaphane a day before. We're going to inject with valproic acid and then continue giving the sulforaphane for a few more days. and then allow the moms to give birth, and we will measure behavior in the offspring. We're going to measure a variety of different behaviors, starting very early on maternal behavior to make sure that the treatments didn't change the mom's behavior. And then we're going to look at maternal potentiation, which measures social interactions between the pup and the mom. We're going to look at sleep in nocturnal versus diurnal activity, because in many cases, autism induces changes in sleep. We're going to look at nest building as a measure of restricted interests. Many times, the mouse models don't build big nests, and marble burying to look at repetitive behavior. And then at 6 weeks, we'll again assess nest building to test whether that behavior actually develops a little later. And we'll test social interaction using the social interaction test. All of these tests are standard tests that are used in this model with VPA. After we do the behavioral assays, we will homogenize the mouse cortex because that part of the brain is most similar to the mini brains that Karen will be studying. I'm interested in a bunch of different parts of the brain, and we'll be sampling different parts, but this proposal is going to fund these cortex analyses. So in both the brain spheroids and the mouse cortex, we will look at antioxidant capacity, total reactive oxygen species, as well as macromolecule damage associated with oxidative stress. What I believe we're going to see is that in wild type animals that are provided with valproic acid and sulforaphane, we should see baseline measures of the different behaviors, and oxidative stress should be similar to the negative control. So all of these should be similar to no VPA. And if sulforaphane's mode of action is NRF2, then the knockout individuals that are exposed to valproic acid and sulforaphane will not be rescued. They will not show baseline levels of behavior or oxidative stress. So that was work that sort of began at ECU that I'm bringing here with me at Harbor Branch. But I also have developed a new research program since coming to Harbor Branch, and that involves harmful algal bloom toxins. So this is yet another model to test whether or not sulforaphane is protective. And so it turns out that algae produces a variety of toxins. And so these human-induced algae blooms are increasing these toxins to levels that are not normal and not healthy. Exposure to these harmful algal bloom or HAB toxins can induce a variety of negative effects across a bunch of organ systems. But the conditions that control the nature and severity of presentation, they're not understood very well. So I'm interested in sort of trying to understand what controls the severity of effects that we find due to HAB toxins. And one thing that has been suggested in the literature is that nutritional status matters. And so we've found that high-fat diet, and by we, I mean scientists, not me, have found that high-fat diet exacerbates renal damage by microcystin. That was done in the rodent model, in the rat. It is also known that antioxidants can protect against a variety of negative effects induced by microcystin. And I should tell you that microcystin is a type of toxin that is produced by blue-green algae. And so these microcystins induce negative effects, and antioxidants have been shown to protect against microcystin exposure. In fact, sulforaphane protects against microcystin-induced toxicity through NRF2. And this has been shown in rodents and mice, and also in 3 different human cell lines. So with funding from the HBOI Foundation, I am testing the hypothesis that sulforaphane can protect against the effects induced by high-fat diet, microcystin exposure, and their interaction. So we are exposing mice to high and low fat food with microcystins, sulforaphane, the combination of those 2 treatments, and a control. So this allows us to get at this generality of sulforaphane's protective effects. And it also allows us to ask specific questions about microcystins. And so we can ask if sulforaphane helps reduce microcystin concentrations by increasing microcystin excretion. We're also interested in oxidative stress and damage, as I mentioned earlier. And we will do histopathology on the brain, liver, kidney, gonad, and gut. Dr. Esiobu and I are also taking samples so that we can study how the gut microbiome is affected, and if sulforaphane is functioning as a probiotic, and if that's playing a role in sulforaphane's ability to protect. Now, one of the problems in Florida is that exposure to Hab toxins is not well studied. And, you know, it's because Hab toxins are highly variable. There's all different kinds of algae that cause all different kinds of different toxins to be released. And so one of the things that we're doing through the Center for Coastal the Florida Center for Coastal and Human Health are to quantify harmful algal bloom toxins in aquatic areas where humans frequently visit. So areas where they recreate or go and visit. We want to look at 10 different sites, and 6 of these are going to be nature centers that we engage in educational programming with. And then 4 of the sites will be around Lake Okeechobee. We want to sample these sites every 2 weeks for 6 months to try to understand where the HABs are, where the people are, where the HABs are, and over what time period we see different harmful algal bloom toxins. And so we can measure for a variety of different types of toxins found across different harmful algal bloom types. In the future, we would like to measure human exposure to environmental toxicants in these same areas. So the idea is that we're going to go out into the community and we're going to measure these chemicals. And then where we find chemical exposure, we're going to go back out into the community and talk to people about what's out there and talk to them about the effects that these chemicals could have and how we might protect ourselves. And then we would hope to engage people in a study that will allow us to measure their exposure to environmental toxins. So through their blood, urine, feces, and potentially through armbands that will collect contaminant concentrations. And we'd also like to conduct a clinical trial to test how sulforaphane supplementation affects toxin concentrations, oxidative stress, and biomarkers of health and disease. Sulforaphane supplements are sold at the vitamin shop, at Amazon.com. So these supplements are on the market already. So it's not, people are already taking them, and they're been shown in many different clinical trials to be safe. So this isn't, too crazy of an idea. So with that, I want to just point out that there were really a lot of people at ECU that I should acknowledge. Tons of undergraduate students and graduate students were instrumental in the hypospadias stuff that I talked about. I've had a variety of different opportunities to earn funding, and there are many little grants from ECU that really helped sort of mend projects together for undergrads. And so those were great. And now I have a new acknowledgement slide, and I wanted to acknowledge the people that I'm working with here at FAU. And I have no idea what time it is, so hopefully there's time for questions.
Okay, Krista, thank you. That was terrific. And yes, we do have time for questions. That worked out very well in terms of time frame here. So I only have one question, which was, what is the name of this seminar? What was the title of this when you first started?
Well, let's go look. How's that sound?
Yeah.
Oops, sorry. I don't know why that went away. Can you see my screen?
Now we're there. Yeah.
Exploring the mechanisms of resilience that reduce the effects of environmental chemicals.
Yeah. So I have a question on, just for all this. Oh, actually, okay. Another one came in. So let me ask this before I ask mine. Okay. Have you looked at whether any vinclozolin, transgenerational transcription effects are reversed by sulforaphane, or is the effect limited to time of exposure?
Can you read that question to me one more time?
Okay, so have you looked at whether any
Vinclozolin?
transgenerational transcriptional effects are reversed by sulforaphane, or is the effect limited to time of exposure?
Okay, that question is really awesome. So I did not talk about the transgenerational effects of vinclozolin. The reason why I didn't, and when I said I didn't talk about it, I haven't studied it either. But that is such a great question, because what we know is that vinclozolin induces changes in the germ cells, that epigenetic changes that are inherited. And so that work is done when the gonad is developing, when the especially, and it's very pronounced in males. And so when the gonad, when the testes and ovaries are developing, and their germ cells are altered epigenetically. The timing of exposure that we use in the hypospadias model is after that has happened. So our animals receive vinclozolin after the time that the studies that induce those epigenetic changes expose their animals. Now, possibly our animals would have epigenetic changes, because we start our exposures sort of at a time where maybe there could be such effects. But to answer the question that was asked, what we would need to do is, rather than focus on causing hypospadias, we would focus on causing those epigenetic changes. So we would expose earlier and expose to vinclozolin and vinclozolin, plus sulforaphane, and ask if those transgenerational effects are different. And we haven't done that, and that would be really super cool to do. Actually, I would love to do that.
Okay, very good. Okay, so another question, still have a little bit of time. Are there any inhibitors of NRF2, and does NRF2 knockout sensitize cancer cells to chemotherapy?
So I'm sure that there are inhibitors to NRF2. And I would have to look that up. And if the person is interested in chatting in more detail, I can, we can talk in more detail about that. The NRF2 is very interesting. In some isolated cancers, NRF2 helps the cancer grow because it reduces oxidative stress within the cancer. But other studies find that NRF2 prevents cancer and also prevents many of the toxic effects that are caused by chemotherapy. And so sulforaphane can reduce a lot of the side effects induced by chemotherapy without affecting the chemotherapy's ability to kill the cancer. And so more work needs to be done to look at the link with cancer, but it's complex.
Okay. All right. So next question. How can these results be integrated into medicine for the treatment of these diseases?
Yeah, that's a great question. So sulforaphane is already on the market. There's already a clinical trial being done on pregnant women with sulforaphane to look at whether or not it helps reduce the risk of placental insufficiency and high blood pressure in moms, which is the name of that is escaping my brain. And so there are clinical trials being done in pregnant women now. They're not looking at hypospadias. They're not, measuring contaminant loads in the mom pre and post exposure to sulforaphane. That would be what I would start with is, to get with the people who are doing that research and ask them to take samples so that we could look at in these women who are getting this through their clinical trial, do we actually see a reduction in toxicant loads? I've thought about many different ways that we can start to translate this work into humans. Another one is to just give healthy people sulforaphane. And so this is something that I would like to do around Okeechobee and the Indian River Lagoon, because I guarantee you that we all have toxins in our bodies. And so what we could do is measure everybody's exposome and then provide these supplements that are on the market and then watch whether or not those chemicals become reduced over time. So that would be another way to sort of get this into human health relevance, I guess.
Okay, very good. Well, that's the end of our questions. We're also at 5:01. So I guess that will be that'll be the end of our seminar presentation. Again, thank you very much. That was terrific. Glad to have you part of our seminar series, and certainly glad that you're here at Harbor Branch. So good luck with things going forward.
Yeah, I would love to collaborate with anybody who's interested. So jot down my email address, and I'm happy to chat. Thank you for the opportunity to present so that people know kind of what I'm up to.
Very welcome. And again, good luck as things go forward.
Thank you.
All right. So bye, everybody.